Yes, Dr. Assefi, we do.
Okay, great. Hello, everybody. This is Dr. Marjan Assefi. I'm CEO of Marie Curie Science Research Center, and also I'm university faculty. We have written some different books about the nanotechnology and neuroscience. This is one of our books that today we are going to discuss about that with Dr. Farzad Masoudi and Dr. Kai Lovansroki. So in this book, we are going to talk about the integration of nanotechnology and neuroscience. But first of all, we should see what is neuroscience. As you know, understanding the dynamic complexity of nervous system is a research challenge in the world of science. And the analysis of the nervous system includes several levels, from the cellular and molecular domain to the systemic, computational, and cognitive examination of the brain. So the specific topic that from the main focus of neuroscience research will challenge over time. And data and result will be added to which increasing the knowledge and creation of new methods and technologies. So developments in electron microscopy, computer science, electronics, functional neuroimaging, biological science, and also genetics all have been major drives of progress in neuroscience. So as you see, neuroscience is the most important major in the world, and the scientific study of the nervous system increased significantly in the second half of 20th century. The main reason for which is the development of molecular biology, electrophysiology, computational neuroscience, which enable neuroscientists to study the nervous system from various aspects, such as the structure of brain, how it studies its performance, how it grows, and inappropriate performance and its changes. So, for example, it's now possible to understand the details of the complex process that occurs in neurons. Nerve cells are specialized to communicate with each other through the cell in the body. They transmit electrical or electrochemical even signals from one cell to another through special connections, which is called synapse, and can communicate with nerve cells and other tissues in this way. So many nerve cells have a thin filament called an axon that may reach a distant part of the body and is capable of rapidly carrying electrical signals. As a result of this connection, the signal transmission from the axon affects the activity of other neurons, muscles, glands, or at the axon termination points. A nervous system consists of a set of neurons connected to each other. And as you know, modern education and research activities that we had in neuroscience can be divided into several branches based on the topic of the scale of the system that we investigate. So this is one of the most essential part that we should keep in mind. And also don't forget about nanotechnology. Nanotechnology is very essential nowadays for us, and it's a manipulation of matter on near atomic scale to produce new structure, material, and devices. And technology promises scientific advances in many sectors such as medicine, consumer products, energy, chemistry, in any other field that you can imagine. And there are the terms of nanotechnology first used by Norio Taichi, a professor at Tokyo University in, I believe, 1974. He used this word to describe the construction of precise material devices whose dimensional tolerance is in the range of nanometers. And in 1986, this word was redefined and recreated by Eric Drexel in the book titled Engine of Creation. The beginning of the era of nanotechnology, he studied the terms in a deeper way in his doctoral thesis, and later developed a book titled Nanosystem Molecular Machines, How to Build Them, Their Calculations. And translated word by word for nanotechnology means very small. Nanotechnology meaning very, very small in the scale as 10 to the negative power of 9. So is the technology of changing property of molecular that make up the material. And for this reason, the nanoscale is the best definition of this technology. Any technology that you can imagine is made up by nanoparticles even. So one of the, there are two approaches for manufacturing of nanomaterial. One is the top-down approach, which involves the breaking down of large pieces of material to generate the required nanostructure from them. And also the other approach is bottom-up approach, which implies assembling single atoms to molecules into a larger nanostructures part. But let's go to the, so when we want to mix nano and nanotechnology and neuroscience, we're gonna have nano-neuroscience. Let's see what is nano-neuroscience. It should be very amazing, is that it? So let's just go over it. Nano-neuroscience is a kind of visualization of neural activity, which is a key importance in the study of neuroscience. And the use of nano-imaging tools with nanoscale resolution are helpful in this field. And also don't forget about these optical image tools include photoactivate localization microscopy and stochastic optical reconstruction microscopy as well, which help to visualize cellular bodies at the nanoscale. Imaging tools have shown that the dynamic and cytoskeleton of actin inside the cells, which was very important to understand how neurons function during the growth of neurons in the response of how the axonal process differentiate properties of receptors, stoichiometry in synapses, and finally, to understand the role of synapses help in the change and dynamic of neural activity. And also, as you know, researchers have focused on devices to stimulate or inhibit neural activity, but the importance aspect of conducting this research is the ability of the device to monitor neural activity. So a major aspect to be improved in nano imaging tool is efficient light collection, because a major problem is that biological tissues are sparse media that do not allow direct propagation and control of light. So these devices use nanoneedles and nanowires for probing and stimulation, which is really important part of nano-neuroscience. So as you see how it's important for us to discuss about nano-neuroscience and nanotechnology and the how they work and how they manipulate. So these are the most important parts that we should keep in mind about that. So let's go to the next slide, which is applied neuroscience. What is applied neuroscience? As you know, the translational neuroscience use basic neuroscience research to translate, develop clinical application, and also treatment for disorders of the nervous system. So this field includes branches such as deep brain stimulation, brain-computer interface, and neurorehabilitation, and the creation of sensory nervous system devices such as hearing implant, retinal implant, and electronic skin. So in terms of research, applied neuroscience is divided into several stages and categories that using a 5-layer system, and one consider a linear progression from basic science to public health application, and also applied neuroscience is viewed as a cycle, which is the public health requiring basic science research followed by public health and clinical implementation. So like basic scientific research, preclinical research, clinical research and clinical neuroscience, clinical application, and general hygiene, which is the most important part. So let's go to the other part, which is electrophysiology. And as you know, electrophysiology be used in the applied neuroscience as a tool. It's a kind of tool to study the electrical properties of nerve cells in animals models, as well as to investigate nerve function disorder in humans. Technique used in animal models, such as patch clamp recording, are used to investigate how neurons respond to pharmacological agents. Both electroencephalography and magnetoencephalography are used to measure electrical activity also in human brain. So for neurological disorders in diseases such as epilepsy, and in research setting to investigate different differences in electrical activity in human between normal and to be used as a nervous system. So let's go to the other part that we discuss in this book is neuroimaging. So neuroimaging includes various techniques used to observe the activity or structures of the nervous system and brain. So positron emission topography, which is called PET, has been used in animal models such as mammals and non-human rodents to identify the target of the molecular mechanism of neurological diseases and investigate the impact of drug addiction. Function magnetic resonance imaging is used to investigate the neural mechanism of drug addiction and the neural mechanism of mood disorder, anxiety disorder in the elderly ones, and the neural mechanism of disorder in such as schizophrenia. So these are some parts that we discussed in this chapter. And also the most important part is gene therapy. Gene therapy involves the transferring part of the nucleic acid to the patient for the treating the defective genes and reduce its effects. For example, in applied neuroscience, the gene therapy is also used to treat neurological disorders. So we can use gene therapy to cure so many diseases that related to the genetics, and have been proven to be effective in treating variety of disorders like neurodegenerative disorders, such as Parkinson's diseases, Alzheimer's diseases, and in primary rodent models and in human, through the use of neurotrophic factors such as nerve growth factor, brain-derived neurotrophic factor, BDNF, which is the glial cell derived neurotrophic factor, and through the use of enzymes such as glutamic acid, we can usually use to transport them into the cell from adenovirus, and it is used as a vector, which can be really essential for people who are using gene therapy. So let's go to the next one. So when we mentioned neurodegenerative disorder or neurological deterioration is the disorders that result of the loss of nerve function. This is really important, but it's happened over the time, not just suddenly, not just immediately happen. It happened over time. So which leads to the cell deaths. And we can see the example that we have with neurodegeneration disorder include Alzheimer's diseases, Parkinson's diseases, Huntington diseases. These are all the diseases that applied neuroscience is investigating the molecular mechanism of these disorders, and also drug delivery mechanism to treat these disorders. And also we use drug delivery system in these disorders to take, for example, medication to the cancer cell and to destroy that cancer cell, but not destroy the healthy cells. So these are the most important part of the drug delivery function as well. So investigating the effect of the blood-brain barrier on drug transfer and its role on the body immune system in neurodegenerative disorder, which is the essential part of it. So when we go to the protective effect of nanoparticles and treatment of a nervous system, these are the important part as well. So as you know that regenerative medicine is a branch of modern medical science whose purpose is to cure the cells. So regenerative medicine is a branch of modern medicine science whose purpose is to repair and restore damaged or lost tissue, organ, or even cells. To the type of the treatment approach and methods include all cell therapies. For example, treatment using the patient's own cells, treatment using cells non-autologous donors is treatment with the growth factor, use of recombinant proteins. So as you see, nanotechnology is one of the new and very practical branches that has attracted the attention of researchers and also created many more opportunity for progress in medical science. For example, in medical science, technology is also used in military agriculture, diagnostic methods, and magnetic imaging, sensors, and quick detection of material. So because of that, researchers use the technology to diagnose and treat many diseases, including cancer. So in that recent years, this science has been accepted as an important branch that has made tremendous progress by focusing on molecular methods. So in nanotechnology, we have the use of atoms and molecules, and it is possible to make material that are used in surgery, dentistry, all kind of experimental science. Although there have been many studies on the effect of nanoparticles, and scientists still do not have complete control over the extent of its effect. In the discussion of imaging method, less invasive methods are more suitable. So nanoparticles are ideal candidates for the development of such imaging methods, as you see. And they are engineering material that communicate with biological system at the molecular level as well, and can create physiological response and nerve stimulation while minimizing side effects. And also nanoparticle, unlike the usual systems such as table solutions, and also intellectually control the maintain of distribution of drug in different parts and cause more and more and better effects. So in this part, we have nanoparticle, which is used in imaging in neuroscience, as well as investigating of the fate of adult stem cells in nervous system for treatment of many nervous system disorders that we have nowadays. And many researchers are conducted a small material that can be used through various methods, such as antioxidant effect, stimulation, and proliferation of nerve cells. I believe that we can cure modulation inflammatory factors as well. So the aim of the study is to investigate the protective effect of nanoparticle in the treatment of nervous system, which is really important nowadays. So let's go. And here in this part, we have medical nanorobots, which is the essential one and the important one that we have. And using the knowledge of nanotechnology, researchers have been able to design biological nanorobots that are placed in human body and play the role of protector and healer. And the smart micro machines are able to make several copies of themselves and replace worn or damaged tissue. And the process is called self-replication. So in the future, we will have prevent humans from contracting all kinds of diseases. They will even delay the process of human imaging, which is really, really essential for us, even I think for all the women's. So we don't want to be aged. So this technology is really good for us. Using the knowledge of nanotechnology, we have been able to design biological nanorobots that are placed in human body, and they can play a role as a healer. So these small micro machines are able to make several copies of themselves and replace all over the tissues. And extensive research is being done in the field of treating diseases such as diabetes, heart diseases, AIDS, and nanorobots can have potential facilitates that are able to protect the system and by community and by placement in colonies. So they are placed in the atomic and molecular structure in known process to complete the cycle. So these are the most essential part of medical nano robots. To design a nanorobot, scientists use neutral models such as the structure of DNA strands. By using the knowledge of nanotechnology, scientists are able to make biosensors in 1 billion dimension. The nanorobots that are made in research centers are so small that are thrown out with dust with sneezing. And the cell was equipped with a fork and several sensors. The sensors were installed in order to prevent collision with obstacles, and the fork's task is to take the sample from the intestinal surface, and the micro machine could either move by itself or be moved by a doctor with a manual control. So the working of the style of the nanorobots that are placed inside the body is simulation of the environment in 3-dimensional space, and analyzing the information in them is based on quantum methods that we might get. So let's go to the energy sources for nanorobots. So how the energy is coming from, how the energy sources is coming for nanorobots. The most effective way for nanorobots to be constantly moving and acting, which is the essential part to move, is to create sources of energy that can be understood from environment in which the nanorobot is working. Fluid kinetic energy, electromagnetic rays, which are emitted from light, are suitable option, I believe, for energy source, which these rays are used for open workspace. Also using temperature changes or increase or decreasing light are also good options. So we should keep in mind that a wide working space, the energy produced from a vibration will be more suitable, which can be used in different environments and including the biological environment as well. So this is the important section of the nanorobots. And we have some advantages of the problem and problems of nanorobots. We can say advantages or disadvantages. In the general, there are two important issues in making a nanorobot. First is that simulation and design based on macro robot, which includes driving force, communication, and orientation, and secondly, assembling nanorobot parts. This is the most important and most difficult part to assemble nanorobot parts. In the field of nanorobotic has a theoretical aspect, and so far no artificial, non-biological, non-robot has been made. But theoretically, advantages and problems have been noticed, which are with the durability of nanorobot, which theoretically lasts for years, their performance time is very short because their movement is limited, and the biological events occur at the same speed but in less time. It is possible that nanorobots will not be recognized by the body as a pathogenic agent due to their small sizes, and the small size creates limitation for sure. because the ultra small size allows the nanorobot to pass through any unknown and unwanted structure without being guided or stopped. So these are some important parts or important advantages of nanorobotics. So as you know, we use nanorobotics for nanosurgery as well. So classical and surgical methods are macro, but some special surgeries such as eye surgery require smaller and finer tools. Therefore, microsurgery emerged, and in the last few years, miniaturizations has made many advances in surgery. The first advantages of nanosurgery is the reduction of the shocks and wounds caused by classical surgery. Also, there are no slips and incision in nanosurgery. These issues make the patient recover faster. So conventional surgeries are heavy and exhausting, both for the patient and for the surgeon. On the other hand, the patients suffer from fear and anxiety. Many patients are prolonged recovery time, and on the other hand, surgeon needs to be concentrated for a long time to perform the surgery completely. So this is the issue. But in nano-neurosurgery, the surgeon uses handler bars to control the arms of the nanorobot, which are equipped with a small surgical instrument, and move them to the desired location. So the other arm of the nanorobots carries a camera that allows the surgeon to observe the environment inside the body. So you see that how much is easier right now to do the surgery rather than past decades. When we come to the finite element method, is a popular method for numerically solving differential equations arising in engineering and always mathematical modeling. Problem arises of interest in including traditional fields of structure analysis, heat transfer, fluid flow, mass transport, and electromagnetic potential. And is the software used in this thesis is that the COMSOL emulator software, which I will explain briefly about the history and working method of this software in next slide. So as you see FEM, which is, we are going to mention it right now about that. So the most important part is that they can interact with other engineering software such as Castia and MATLAB, and also R. They can use with R as well. So the R is important part, and it is also gives an user an opportunity to analyze and simulate a sample structure in a form of mathematical formula, in the form of equation and physical ones. So if FEM subdivides a large system into smaller and simpler parts called finite elements. So this is achieved by a particular space discretization in space dimension, which is implemented by construction of mesh of the object and the numerical domain for the solution. Part. So this is the most important part of it. And also strategies to understand them, to mean a clearly defined set of procedures that cover creation to finite elements and definition of basis functions. So these are some softwares that we can use for nano-neuroscience, like FEM, like COMSOL. These are some softwares that we might use. And when we come to the part of the COMSOL software at the following physical capabilities that can be applied to different parts based on their equations. So as you know, the console physical, they have physical capabilities, different modules they use, and this software has been marked by console, the latest version of which is official version, which released in 2013. And this software can be installed in Linux, Windows, and Mac operating systems, same as FEM that we used. And for the FEM software, which we talked in last slide, it's some program can interact with engineering stuff, such as MATLAB that I mentioned. It also gives user the opportunity to analyze a sample structure, the form of the mathematical formula for neuroscience. So, so as you see, we used so many softwares for our neuroscience part. But the most important part before the conclusion that I want to mention is that we forgot to mention about the common sabot artery. There are two common cardioid arteries, or the right and left common cardioid arteries. Each have created smaller and called the internal cardioid arteries, which supplies blood to the anterior region of the brain. So this is the most essential part that we should focus more in the neuroscience field. And the origin of the right and left common sabot arteries is not the same. The right common artery branches from the branchial or innominate arteries. The branchial arteries is the thickest branches of the aortic, which is 4 to 5 centimeters in diameter behind the right stroboclavicular joint. It divides into two different branches. And so in the internal cardioid artery is one of the most important artery that head and neck, which branches off from common cardioid arteries, which we had. So, and one of the most important part, that two region of the arterial cerebral artery, and also which is area before artery communication, and also area after the artery communication. These are the most essential parts that we should talk about these. And also in the case of blockage of the common Cobb artery, all the signs and symptoms that occur when the internal Cobb artery is blocked may be observed. And sometimes there is a possibility of blockage in the origin of left common Cobb artery, left subclavicate artery, and in which case we can refer to the pulseless diseases or artio-arc syndrome. So there is occultation of the great arteries separated from the aorta. In fact, that inflammatory diseases of the aortic arc that leads to the blockage of aorta, and its main arteries and characteristic of this category of diseases. So with all these symptoms of all these diseases, so we see that we can use nanotechnology to cure and to prevent all these symptoms, which is really essential. And diagnostic is listening to the sounds of the cardioid artery, and also near the angle of the lower jaw is diagnostic method due to the creation of the stenosis area. And also the treatment, it's based on the whether the disease is systematic or asystematic. The treatment methods should be medical or medicinal method, endarterectomy or surgical removal of fatty plaque from the inside of the vein, which are done by nanotechnology. So these are the most important parts that we can do with all of these with nanotechnology parts. And also we can manufacture the non-viral drug nanocarrier for even cancer treatment. So as you know, the scientists succeed in making non-viral drug nanocarrier, which are made of polyethylene and have nanometer dimension and can be widely used in pharmaceutical industry, especially for the treatment of cancer. So as an association with the cell targeting molecules with gene nanocarriers, in addition to keeping gene fragments around the cells and facilitated their entry into certain cells, prevent them from entering non-cancerous cells, and prevents the occurrence of side effects caused by aberrant gene expression in healthy cells. So the aim of these drug delivery is to present project where was to synthesize the polyethylene amine derivatives with different molecular weight and attach cell targeting ligands on them, a new series of polymer and based system in nucleic acid delivery. So first nanocarrier based on cationic polyethylene polymer were prepared, and then their physical properties such as the ability to store the genetic parts, particle size, and surface charge, the ability to cope the environment changes were investigated. In the following parameter were attached to the cationic, as we mentioned, to the polymer as a molecule targeting the surface indicator of cancer cells and the ability of the final nanocarrier to transfer gene fragments to, and also to prevent their entry into cell without cancer indication, which is one of the most important parts of it. So researcher was conducted prostate cancer cell using 14 different nanocarriers with different structural modification, medicines, or gene fragments combined with this class of target of nanocarriers will be able to stop growth of cancer cells and cancer, including leukemia, with the help of gene responsible for regulating cell cycles, and pointing out that these nanocarriers will reduce the side effect of caused by aberrant gene expression in healthy cells. And also, don't forget about that such carriers have the ability to enter to the pharmaceutical industry as a new generation of drugs after undergoing the clinical studies. So in conclusion, we are coming to this point that in this book, we explained neuroscience, difference between neuroscience, nanotechnology, and if we mix nanotechnology with neuroscience, which is nano-neuroscience, and all these treatments and all the software that we use in nano-neuroscience branch, and we have taught the softwares, how to use those softwares, and how these technology can treat the important diseases, and even how to prevent aging, which is essential part nowadays. So all these stuff discussed in this book. So thank you so much for your listening. I really appreciate. So this book is written with me, which is Dr. Marjan Assefi, and Dr. Farzad Masoudi, and also Dr. Kai Lovans Rocky. So we will be so happy to hear your ideas. Thank you so much.